So this is the Unit 1-2 review, and I'm just going to go through the essential questions and answer them. So the first, well, these aren't essential questions, but the first thing you need to know is remember there's the bacteria structure. So first there's a capsule outside the cell. Now it's not shown on this picture, but where it would be lining this outside of the cell, and can be found on gram-positive or gram-negative bacteria, and it's a sticky substance that allows bacteria to stick to things. The cell wall is the next layer, and remember gram-negative show up to be pink, so if I highlight that, that's all gram-negative, and the layer that I'm actually highlighting there is the outer membrane, which is the lipopolysaccharide membrane. And remember that gets washed off after we stain it purple, and then when we stain it pink, this peptidoglycan wall right here gets stained, and that's why it turns pink. Whereas the gram-positive turns purple when we stain it, and it has that thick peptidoglycan wall here. Endotoxins are only found in gram-negative, and they're found right in here in this lipopolysaccharide membrane, and they will kill surrounding pathogens. And then finally we have the cell membrane in here, this inner layer right in here on both cells, that controls what comes into and out of the cell. Furthermore, with structures, we have, I have to clear my page here, is we have a nucleoid, which is the main cellular DNA. Chromosomal DNA is the thick region part in here. Then we have the plasmid DNA over here, and this could be, there could be several plasmids in here. In this case there's one. It looks like there would be one right here too. And they are just circular pieces of DNA that often have anti-bacteria resistance genes on them. We all know what ribosomes are. They're found all over throughout the cell. They make proteins. We got the flagella back here. It's used for movement. And then the pillae all over the cell that is used for exchange of DNA and also attachment to other cells. Alright, now finally one more thing before we get to the essential questions. We got our antibiotics. We got our sulfa that inhibit folic acid. We have our quinolines that inhibit DNA replication, so our bacteria can't replicate. We have our penicillins, which are our beta-lactams that inhibit cell wall synthesis. And then finally down at the bottom we have our tetracyclines here that inhibit the ribosome, and the ribosome can no longer make proteins. Alright, now on to our essential questions. First one is, how do antibiotics work to fight bacterial infections? The first one is efflux, which I inhibit to like a gag reflex, where the bacteria just wants to get rid of that antibiotics and spit it out. Here's a quick little video. You guys have already seen it, so I'm just going to fast forward. But basically the antibiotics come into the cell and then get actively transported out of the cell and spit out by the bacteria. So the antibiotics do not harm the essential parts like the DNA or the ribosomes. And it's actively transported because remember there would be a high concentration of antibiotics in here, and they'll have to actively use energy to push it out. Then we got destruction or inactivation. That's the bacteria produces enzymes down here, and most of those enzymes could be coded by the plasmid or by the nucleoid DNA itself. And these enzymes break down the antibiotic and make it useless or actually inactivate it down here. These enzymes are surrounding the antibiotic and inactivating so it cannot cause damage. Then the last one we have is our basic mutation in the DNA. So normally our bacteria would be killed, but then after a mutant, our bacteria survives and is all happy and runs amok. Alright, now we got the methods that bacteria use to share antibiotic resistant genes. So you guys all remember the first one, the conjugation. Alright, and that's where we use our sex pili, or our sex pistol in here, that where it connects, and it's a one-way transfer of the plasmid. So this plasmid right here will get copied and put into this cell. So this first this donor would have been maybe resistant to penicillin, and now this cell will be resistant to penicillin because they have this plasmid that made it resistant. Alright, so that's conjugation. The next one we have is transformation, and that's where after a cell dies, you can either get DNA fragments or an intact plasmid, and this bacteria is nice and greedy and wants to pick up the plasmid and put it in a cell, or that DNA, and that DNA could contain bacteria-resistant genes. And the last one is transduction. The virus right here, it starts up here in number 1, and then it goes over, this is where the bacteria infects. So basically the virus infects the bacteriophage, the virus infects the DNA, puts its DNA into the donor bacteria cell. This is bacteria cell. The virus DNA breaks up the bacterial DNA and uses it to reproduce. A bunch of virus babies are made right here, and some of them, these ones with the purple strand, have the bacterial DNA in it. These viruses come over here, inject the bacterial DNA into it. Since the virus DNA is not there, it doesn't infect the cell, but the bacterial DNA gets into the cell, and that could have antibacteria resistance gene on it, and that can be incorporated into the bacteria's DNA, and now it's resistant. Alright, so the transduction is when a virus puts it in to the cell. Then finally, our last question is, how do scientists gather information during a potential outbreak of an infectious disease? So this is a review of 1.1, and basically we use bioinformatics, which we take blood samples of not our DNA, but actually the pathogen DNA inside our body, throw it into the DNA database, sequence the DNA, throw it in the DNA database, and compare it to identify the DNA. We can also do ELISA tests and test for the antigens and antibodies to see if we have that specific pathogen. We can interview the patients and look for connections between them, see what kind of food, see what activities they're participating in, and then finally we can use other diagnostics, which we can take MRIs, CAT scans, and just simple X-rays and other blood tests to diagnose the patient. And that's it.